All right, good morning, everybody. We're going to get started. It is my great pleasure to introduce today's speaker, who needs no introduction. We all know Carol very well. Dr. Carol Lange, to give her presentation today on her work on targeting related steroid hormone receptors to combat breast cancer. Carol has been here for a few years, a few longer than me. I actually remember Carol back from her days in Colorado when she was a postdoc there, and then she moved on here and has established a really fantastic lab. She's the world's expert in studying progesterone receptor in breast cancer, and I think has a huge amount of awards and accolades. I know she's very involved in the Endocrine Society as well. From a personal note and her sort of work here, she has always just been an incredibly supportive faculty member, particularly of trainees and of other sort of early career faculty, including women. And so I think a lot of us have really benefited from her support and mentorship over the years. So I'm thrilled to introduce her and have her tell us about her phenomenal work.
Thank you. Thanks, guys. Thanks for making it here. And anyone online, thank you for joining me. So today, I thought I would focus primarily on our work on progesterone receptors. Early in my career, Mark Lipman visited and came into my office, and I don't know if you know, he's one of the very first people who treated breast cancer cell lines with tamoxifen, and is very famous for that work and helping to get tamoxifen into the clinic. But he came to my office and sat down and said, "Carol, why are you wasting your life working on progesterone?" So here I am 25 years later, wasting my life, and we'll see if you think so, we can have an anonymous vote or something. So we do have a program in cortisol, and I'm not going to talk about that today. That would be a topic for another time. It just seemed like too much. So we know breast cancer is incredibly common, 1 in 8 women, rapidly becoming 1 in 7, expected to even become even more common as we deal with the effects of endocrine disrupting chemicals, for example. But the risk factors, many are out of our control. They include gender at birth, female gender at birth, age, genetics, breast density, and we go towards things we can control: hormone exposure, reproductive history, diet, exercise, lifestyle, life circumstances. Stress feeds into breast cancer for sure. And these things all work together and conspire to make this a very common disease, even outside of these things like genetics. And I'm not going to spend too much time on this slide. We know there's molecular subtypes that are defined by the expression of the estrogen and progesterone receptors and the amplification of the HER2 tyrosine kinase. And as we move towards the left and we don't have these proteins expressed, it becomes much more difficult to target these cancers, and they rely heavily on chemotherapy. So today we're going to focus mostly on the luminal or most common type of breast cancer, which is ER positive. And steroid receptors are ligand activated transcription factors. They're actually expressed in all types of breast cancers. There are really no steroid receptor negative breast cancers now. Even triple negative has GR and a subset has AR. But the luminals, we think of PR expression primarily as a clinical marker to signify a functional ER. So we look at the expression of ER and the expression of PR. PR is an ER target gene. So the PGR gene is one gene that encodes progesterone receptor isoforms, and that is regulated by estrogen and estrogen receptors. And so having the two together kind of suggests that the ER is activated. And these are all expressed, as I said, in breast cancer. And if you have ER and PR and GR in your tumor, you have very good outcome in luminal breast cancer. And more recently, we've also been looking at the mineralocorticoid receptor, which is also found in these breast cancers. And so it's quite interesting that now we have this whole family of steroid receptors that we realize we probably need to study. Instead of focusing on one or two, we need to kind of think about this collection because they're highly functionally redundant. They share co-activators. Lots of different interplay happens. And so today I'd like to talk mostly about the interplay of the ER and the PR. But just as one example for GR, for example, this is published work from Suzanne Konzen's lab, where if you have ER positive cancer, untreated and chemo treated, and you have GR, you do really well in the red. You do really well with a lot of GR. But if you have triple negative and you have a lot of GR, that's a completely different story. You do really poorly. So the biology is quite different depending on the context. And that's certainly a theme in steroid receptor biology. So this is the famous hallmarks of cancer wheel. And I just wanted to call attention to are really maybe too centric on proliferation on this wheel. So we say uncontrolled proliferation is cancer, and we look at proliferation. And I would say ER and AR are really well accepted to be drivers of proliferation in breast and prostate cancers. I'd put PR somewhere in the middle. Sometimes it's inhibitory, sometimes it's stimulatory to proliferation. And this is highly context dependent. These receptors can also be immunomodulators. They can mediate changes in inflammation. Certainly GR and PR have been implicated in metastasis in the literature and DNA damage. I think all of these receptors are mediators of pro-survival. And we've found more recently that they can mediate metabolic plasticity. And so these other hallmarks are things that cancer cells can do when they're maybe not dividing or dividing very slowly. And I think we sort of ignore that a lot, maybe to our detriment in our field. And so why should we care about the cell cycle? Why should we care about these activities that cancer cells can do when they exit the cell cycle, or they might be growth inhibited by a treatment, but still alive? And so why should we care about that? And I think what happens is, you can kind of say cancer cells do not walk, talk, and chew gum at the same time. And meaning they don't proliferate and migrate at the same time. They stop proliferating, they undergo EMT, and they become highly migratory and invasive. And they can enter what we like to call dormancy and sort of remain dormant in the body, or very low proliferative index, and give rise to supporting the stem cell compartment and seeding metastases. And so that's why we should care about G0 cells. And how do cells enter and exit the cell cycle? What is that decision like? And so I just wanted to introduce this a little bit. There is a complex called DREAM, and this stands for dimerization partner, RB like E2F MUB DREAM G0. So cells in G0 have an activated DREAM complex. And so transcriptional regulators that coordinate, they repress the cell cycle by activating this DREAM, and it happens during quiescence. And it's about 900 genes, about 900 cell cycle genes, where this activated DREAM is sitting down and repressing the cell cycle genes. And with re-entry into the cell cycle, you can see a shift in these partners that activate the cell cycle gene expression. So you have this stream complex that's very large, and there's some stoichiometry changes. And then basically what happens is you can release it and enter the cell cycle. And for today's talk, just kind of think of it as when DREAM is activated, 900 genes are repressed, the cells are entering G0. And when that repression is lifted, then this other complex takes over, and the cells can enter S phase. And these components are frequently altered in cancers. Their expression is altered. Their expression is frequently altered by viruses that cause cancers, such as cervical cancer. And so it is kind of a regulatory node on whether cells have the cell fate of dividing or being alive in G0. Okay, so one of the themes in my lab, a major theme, is that in the context of these early genetic events that we think about, oncogenic events, activation of signaling molecules, steroid receptors, which are sort of downstream of all that, their functions are altered. And so that causes alterations in hormone responsiveness. It'll cause alterations in transcriptional activity and stability of the receptors. And it changes the promoter selection. It changes the transcriptome. And this results in altered cell behaviors and changes in cell fate, such as engagement with dream, and also cytoplasmic signaling properties are altered. So these receptors have two functions. They're transcription factors in the nucleus, but they're also bona fide signaling molecules. And so we'll talk about that. So we have this big ugly tumor with a very complex tumor microenvironment, usually hypoxic in the middle, high reactive oxygen species, a necrotic core, et cetera, very cytokine rich. And when we try to inhibit this with treatments, for example, we can get cells escaping this toxic environment by going into G0 and getting out of here, using EMT to get out of here, and seeding metastases. And we're going to talk about the ER positive and PR positive role in this process, and the role of PR isoforms today. So here's the stick figure of progesterone receptors. They're highly phosphorylated. There are at least 14 sites. I'm only showing you some mitogenic kinase sites here in the N-terminus. They have a ligand binding domain here. I don't like this little pointer, it's too slow. And a hinge region, their DNA binding domain, and then a large N-terminus that is heavily modified by post-translational events. Again, estrogen induces the expression of the PRA and PRB isoforms. PRB is a long form, and PRA is a truncated isoform that comes off of an internal ATG. And these are usually expressed in normal tissues at a 1-to-1 ratio. And a very early event in breast cancer is that they become very lopsided. Okay. And then a majority of estrogen receptor positive cases, they appear to have sort of an A-rich, PRA-rich. So B can go down, making A-rich, or A can go up, or both can happen. And I'll return to that theme later. And these are phosphorylated by highly mitogenic protein kinases that are often activated in breast cancer. And a lot of work from my lab in the past has shown that these phosphorylated progesterone receptors are hyperactivated at certain genes, and they can be activated by extremely low dose ligands. Okay, and then we focus quite a bit on this serine 294, which is a MAP kinase site in the N-terminus. We actually made a custom antibody years ago, and it turned out to be a really good antibody. We screened a TMA that we got from Kaylee and showed that at least half of the ER positive tumors expressed a decent amount of this phosphorylated PR. And that was usually nuclear and focal, meaning it was activated. And that has become a really useful tool. And just as an example of just how useful that tool is, Laura Morrow, who is a close collaborator of mine, is focusing on high-grade serous ovarian cancer that occurs in the fallopian tube. And she was able to show using our antibody that these fallopian tube samples from patients expressed really good levels of progesterone receptor using our antibody, and that they had phosphorylated progesterone receptor in the normal fallopian tube epithelia. And then she went on to look at these early lesions, these stick lesions that arise in the fallopian tube and later give rise to high-grade serous ovarian cancer. And you can see these really beautifully stained images of total and phospho-PR. And on the right, you see this nuclear focal staining pattern of phospho-PR. And then we were able to show that these guys, for example, in these early lesions have this activated PR with numerous samples. And it led to this really beautiful paper that Laura put together. So we're looking for more samples to stain. We'd love to stain early pancreatic cancer. We'd love to stain lung cancer. These are cancers that have a lot of progesterone receptor. So please contact us if you would like to have some antibody. So getting back to how these actually function. So all these receptors function in both the nucleus and the cytoplasm. And And they sort of rapidly and dynamically shuttle between the cytoplasm and the nucleus. And what they're doing, they're sampling. They're sampling the cytoplasm all the time. They're sampling what are the molecules that are stimulating the cell. And they're just doing this all the time. And so it's been known for many years that they exist at the membrane. About 5% of ER and PR are located at the membrane. And they're actually prenylated up in there in the membrane, and they can activate the MAP kinase pathway very rapidly. And so either progesterone or estrogen will activate MAP kinase very rapidly. This has kind of been known as the rapid actions, and I think very much underappreciated. And they can activate all these kinases in the literature. AR can do this, MR could do this. So now we appreciate that not only are they together, this membrane complex, but they're frequently together when they regulate genes, ER and PR. So I'll be talking about that today. And this, again, I think is quite underappreciated when we treat patients with drugs that block the ER transcription, like tamoxifen. What happens over time is more and more ER goes to the membrane, and you end up with endocrine resistance in which a lot of ER is at the membrane. And what it's doing is talking to these protein kinases, because that activity is not being inhibited. Only the nuclear activity is being inhibited. And so you're selecting for cells that are favoring the cytoplasmic activity. And it's sort of a fringe field. And so not very many people really study this field. And so in our lab over the years, we've done a lot to try to figure out the biochemistry of this. And we've linked numerous kinases and phosphorylation events to these complexes and rapid signaling. And we've also shown that what happens here is this highly specific phosphorylation signature actually determines promoter selection. And so I think this is a step in the pathway. I think that steroid receptors recruit protein kinases in order to get these phosphorylation events, and they're almost like addresses to specific genes. So it's really all integrated, in my opinion. It's not really separate. I don't think this is how you activate MAP kinases. I think this is how you phosphorylate steroid receptors. Okay, so and they cooperate with a lot of other factors when they're phosphorylated, such as AP1 and SP1. These factors are also highly expressed in breast cancer. Okay, so this is kind of the paradigm now, is that these hormones enter the cell. We've shown that what happens with hormone-rich environment is these guys get sumulated, and sumulation is a repressive mark on steroid receptors, and so it represses a lot of genes. But when you have a high kinase-rich environment, what happens here is the sumulation comes off, and basically you have a de-repression of these genes. And so you can think of it like DREAM. It's sitting there on genes, repressing them, and then when phosphorylation events get rid of SUMO, it's free, and those genes get regulated and upregulated. So it's a little bit reverse. We think of genes getting activated all the time, but steroid receptors repress about half the genes and they activate about half the genes. And so this is sort of a de-repression mechanism. And so what the story I'm going to try to tell you today is sort of like this, and how we think about this is these ER-PR interactions are altered in breast cancer, and sometimes they're cooperative and sometimes they're inhibitory. So we need estrogen to get the PR gene expressed, and then it will inhibit growth with the estrogen receptor. So it will be stimulated, lots of PR, lots of ER, lots of hormone. It actually serves as a little bit of a brake on estrogen-driven growth. However, when we have these phosphorylation events and that SUMO mark comes off, now we have a different relationship where they're cooperating in interesting ways in cells that might be in G0. So here's the analogy. We have this estrogen receptor, and she's got her progesterone receptors with her. This dog is obeying. There's some slack in that leash. That dog looks a little worried, right, Jaime? But it's a good relationship. Everything's cool, and then we have this dog that's kind of pulling back and inhibiting the estrogen receptor, and it's a little bit phosphorylated, and it's getting a little wonky there. And then this dog is really pulling the estrogen receptor, and now she's being activated by this dog because it's pulling her. And so, you know, when this dog breaks this leash, what happens, you know? So this is kind of how we're thinking about it, is sometimes it's a nice cooperative relationship in which everybody's doing their thing. Sometimes it's inhibitory to the estrogen receptor, and sometimes it's activating. And how do we figure this out? So one thing we have done in the lab is we use progesterone receptor negative cells that are breast cancer cells that have been naturally occurring and selected by flow cytometry. And into those cells have been expressed either the PRA or the PRB isoforms. And then we can put, for example, mutations to phosphosites or the sumylation site, which is that lysine. And here you can see Noelle Gillis in my lab made this really beautiful blot, and you can see that the CO cells, which have both isoforms, are here, and then you have the isoform specific cell lines, and everything looks pretty good there. And so some of the things we've done in the lab is to look at this interaction using this proximity ligation assay, in which you can use antibodies to each receptor and show that they interact. And you can see, I don't know if you can see the red dots, but you can see that with estrogen or synthetic progestin, R5020, these receptors will interact directly in the nucleus of the cell, and it's quantitated over here on the right. And so we've been using this assay to see how is this interaction regulated, and is it something that we can target and try to understand breast cancer progression with. So we've been doing a lot with just sort of an estrogenized system where we're add estrogen and look at progesterone receptor activity. So often we don't even add progesterone, we're just looking at estrogen and the ability of the progesterone receptor to sort of be a scaffold. And when we did that, we just basically treated cells with estrogen, and we treated cells that either didn't have PR or had PR, and we got this 40 gene signature that were estrogen induced genes that required the presence of the progesterone receptor, but not progesterone. And when we looked at this, this gene signature, it was very dangerous for survival. And we've also done things like narrow down our list using validation assays of phospho-PR target genes. And these are a little gene list we published and patented that is 15 genes that require the phosphorylated receptor that predict poor outcome. And we've also done things with, like, for PDX models. These are 2 PDX models that we got from Colorado from Carol Sartorius. And what's really interesting to me is if you look, I don't know if you can see that, that is the total monoclonal antibody that's used clinically. And if you saw that, you'd say, well, not very impressive. I don't see a lot of PR there. But now look at, and these are, one of them is a ESR1 mutant, the DG mutant. The other one has an amplified estrogen receptor gene. So they have a lot of ER. But then if you use our antibody, that's a polyclonal, you can see quite a bit of phospho-PR. And so we think that these modifications to PR are probably masking the monoclonal epitope that's used clinically. And so we're missing some of these PR positive tumors. And we can also see in this brain metastasis in this mouse, lots and lots of nuclear phosphofocal PR. And then Carol Sartorius did single cell RNA-seq off of these and looked at a couple genes. And she looks at these stem cell genes, and one of them is KLF4 that was increased. Progesterone receptors were quite increased in her gene signature in the brain, and IRS1. And so we got pretty interested in IRS1. And then the IGF receptor actually went down, but the IRS1 adapter protein went up. And we had a whole series of experiments to show that this IRS1 was also playing in this complex. And you can see here with estrogen, you see the recruitment of IRS1 to the estrogen receptor. And if you mutate the phosphorylation site on PR, you lose that. So again, this phospho-PR is bringing in the ER and the IRS1. And some of the things we did to kind of look at this is using this idea that these the phosphorylated PRs become desumylated. We could bypass that regulation by just mutating that sumo site. So we mutated that lysine to an arginine to get rid of that sumo and sort of mimic the phosphorylation. And when we do that, we get tamoxifen resistance. So Those cells with that 1 point mutation are tamoxifen resistant in 2 cell lines. And they also become quite exquisitely sensitive to insulin in this growth curve. So it's kind of like they don't really need estrogen anymore. They really want to have insulin. This is why sugar is bad. So then we also have these increased expression of these stem cell markers just by removing that sumo site. And we get the increased formation of mammospheres, which is kind of, or tumor spheres, which is kind of a sort of an indirect stem cell assay. In the desumulated in yellow, you can see they form a lot more mammospheres, and those are inhibited when we inhibit the IRS1 with a small molecule or the alpha IR receptor, which we think this is going through. And then we started thinking, okay, if we can make them endocrine resistant just simply by mimicking phosphorylation, what's actually happening in real endocrine resistant models? So we started collecting all different endocrine resistant models from collaborators. And here's an example of tamoxifen resistant. They make more primary and secondary mammospheres, so they have more stem cell capacity. If you look at PR, it's really low expressed, which happens, and it's known that in more advanced tumors, you still have expression of ER and PR, but it can be greatly reduced. But we see it. And what's really interesting, it actually gets induced over time when you make secondary and tertiary mammospheres, the mRNAs get induced. So that's kind of interesting. And then if we look at flow cytometry, we can easily see that they contain the phospho-PR and the target genes that we like to look at that we know are in our gene signature are increased. Even though they have very little PR, we can see these phospho-target genes. And these are all known tumor suppressors that inhibit growth in our hallmarks wheel, but they all are stem cell drivers in the literature. And they all mediate cell cycle exit into that G0 that I was talking about. And we can also see the expression of the proteins, not just the mRNA. And then when we knock out the PR with shRNA, we can see fewer mammospheres, and we can target them also with this IRS1 inhibitor and the PR antagonist onapristone. You can see that one of the cell lines, the T47Ds, are pretty resistant to onapristone. They have a really activated AKT pathway, but if we switch to mifepristone, it works a lot better. It's much stronger. Okay, then we got these letrozole and fulvestrant resistant lines from Tony Diosoro at the Mayo Clinic. And we can see Caroline Deeb did this with flow cytometry. They have phospho-PR. If you do a Western blot of these cells, you can't see it. But if you do the flow cytometry, it's very, very easily visualized in the letrozole and the letrozole fulvestrant. And if you knock it out, you can see smaller tumors. So Tony went ahead and knocked out the target genes of PR, KLF4, and STAT3, as well as PR itself. And all three of those knockouts resulted in many fewer smaller tumors. And he quantitated them here. And he also did this at 5,000 cells and saw the same thing. And then finally, another endocrine resistant model are these ESR1 mutants. And so women on aromatase inhibitors often have, a small percentage of them at least, get these mutations. And these mutations arise under the pressure of aromatase inhibitors. And you can see all the different ones, and they're all sort of in this ligand binding domain. And what they do is result in an activated estrogen receptor in the absence of estrogen. And so it's a way to circumvent the need for estrogen. And they have been a clinical problem. And so Tu Trong in my lab and friends, I didn't know how many friends you had here, quite a few, right? Got a hold of these, and we got some models from different collaborators and looked at PR. And what's interesting is in this set of experiments where she immunoprecipitated the progesterone receptor and found that they do not interact with the ER. So now the dog is off the leash. They are not together. You can see the wild type there on the left where she pulls down the interaction and it's completely gone in these mutants. And we thought that was interesting. She also confirmed this very nicely with PLA. And so this is a very much reduced ER-PR interaction. And then we thought, okay, so what's happening here? And so in this example, these cells are making more stem cells. The Y537S in pink makes a lot more tumor spheres. And when you treat with different hormones, it doesn't really matter. And then you can come in with SERDs and SERMs that target the estrogen receptor, and you can get a reduction here. And she used tamoxifen, elacestrant, and lasofoxifene here, and in two cell lines. And then she has the tumor spheres as well. And then also repeated all of that in MCF7s, and it repeats very beautifully. The other thing that we found is that these models, even though we think this is progesterone receptor driven, they're not very sensitive to antiprogestins. So here we have mifepristone, which is also called RU, or 027, which is another onapristone-like drug, and onapristone. And it's very modest. You see some, but not much. And then 2 went ahead and knocked out the progesterone receptor. And then she saw a very dramatic reduction in the number of tumor spheres and the soft agar colonies. So they are progesterone receptor driven, but they're not very responsive to antiprogestins. And then one thing that she's also done is we've used this mammary intraductal injection model that was innovated by Catherine Briskin, where instead of doing tumors in the fat pad, which isn't really the microenvironment that breast cancer forms in, Catherine Briskin developed this technique where you inject right into the nipple, and that gives you a 3D structure and a hormonal milieu that much more supports these the growth of these tumors, and they look a lot more morphologically correct. And so the field has been kind of switching to this model. And when 2 did that, really interestingly, when she knocked down the progesterone receptor, you can see in the wild type, they actually give you a larger tumor. And then it's not significant in the mutant. However, if you now collect the blood and look at the circulating tumor cells, you see that in the Y537S, there are many more circulating tumor cells, and that the knockdown of the progesterone receptor in the mutant, in the ESR1 mutant, reduces that. Okay. So 2 has also done with other helpers in the lab, Alpe Temes and Noel Gillis, is to look at the differentially expressed genes in these ER mutant cells. And what's really interesting is she sees this really nice difference of genes that are altered with estrogen versus progesterone. And then in the mutant, they're no longer responsive to estrogen at all on the right. You don't see any pink. They're just responsive to progestins. So these cells are highly responsive to progestins, but not estrogens, of course, because the ER is activated in here. And one of the top gene sets that she found was this unfolded protein response, as well as hypoxia, which is a cellular stress pathway. The unfolded protein response is also a really important cellular stress pathway. And it's this pathway in which you have this complex pathway where proteins are misfolded. And the misfolded proteins can activate these receptors that can often lead to cell death or a pause in proliferation. And there are inhibitors of the pathway over here on the right that we've listed that 2 collected and was able to look at those inhibitors. And she found that those inhibitors also have kind of a modest effect. They lower it, that this is tumor sphere assays, so it's looking at the stem cell compartment. They lower it just a little bit. However, when she tips it to be more active with this drug URSO is an unfolded protein response activator that has been developed by David Shapiro as a breast cancer drug. And now it's almost like these cells are hypersensitive to hyperactivity of the UPR, and they undergo death. And you see this really dramatic effect on the tumor spheres And the PR is no longer phosphorylated in the presence of URSO. So it's blocking that PR phosphorylation site that we know is important for this biology. Okay, how are we doing? Questions so far? Okay. So excuse me. So I just wanted to show you this little vignette that Noelle has been working on in my lab. Noelle is a postdoc in my lab who's extremely talented. And she has taken a closer look at the isoforms in this biology that we're talking about, the stem cell biology. And looking at this, we can see that if we take our PRA cells and our PRB cells, the PRB cells are more proliferative. They're better at proliferation than the A. Okay, so the PRB cells proliferate faster than the PRA. But if we do a sphere assay now where we strip out the steroids from the medium and add back progestins, we can see that the A cells are really the drivers of the stem cell compartment. And Amy Dreyer published this quite a while ago in my lab, but we've never really taken a deep dive into the mechanism of this. And so Noelle wanted to repeat this in her hands, and it repeats beautifully because we had never stripped out the steroid receptors before to really show the dependence on progestins. Okay, so now PRA makes more spheres than PRB. And then when she looked at our mutants here, you can see that, again, the PRA dominantly drives the tumor sphere expansion in 3D relative to B, and that we need that serine, that phosphosite. If we mutate that SUMO site and open up the receptor and let it be activated, you get a lot more of the spheres. And so it sort of establishes that this phospho-PRA is sort of the dominant driver of this activity. She also put them in this MIND model in mice with the help of Angie's chemo brain. Sparts. Sorry, Angie. God, it's true, chemo brain. So anyway, if you look at the tumor growth, they're pretty similar. Again, we're so focused on tumor growth, Ki67, but the stem cell compartment is pretty different. When you collect the blood of these mice and try to grow it up, you see that the A gives you more circulating tumor cells. So that made sense. And so then Noelle went ahead and did the RNA-seq. And we have this really interesting idea or thing where the PRA regulated genes happen very dramatically in 3D. The PRB regulated genes are really happening in 2D. So in 2D culture, PRB is more regulated by progestin, but in 3D suspension, where the cells are forced to be in suspension, really PRA is being regulated. And we thought that was quite fascinating. And so you can see that when you wipe out that phosphorylation site, you lose quite a bit of those genes. And really interestingly, I think, and this is a first, is when Noelle looked at this in 3D for the PRA signature, it really tracks with poor outcome, with endocrine resistance and poor outcome. And I think that is the first demonstration that the PR signature isolated from PRA cells in 3D. That's never been done, but I was actually really surprised to see this huge difference. And if you look at what those genes are, they're all cell cycle genes. They're all things predicted to inhibit the cell cycle. And so then she looked at ER and she found that ER and PR are together often at these genes. And so again, we're back to the ER, PR being together. And then she looked at where these are, where ER and PR are together. And she found that they're more likely to be at these super enhancers. And some of the gene ontology included things like TGF beta signaling, cell cycle checkpoint control, mitosis, positive regulation of cell proliferation, et cetera, cell mobility. And she then isolated these, sort of looked at these 5 sites that were highly correlated with both ER and PR being present. And she did this really cool trick where she put in these guide RNAs that sterically hinder these super enhancer sites. And so the ER and PR cannot sit down. And when she did this individually, every single one of them sort of wipes out that sphere growth assay readout. And so all of these nodes where we have ER and PR at these super enhancers seem to be required for this biology. And then she's gone through some of the clinically used anti-estrogens and anti-progestins, and she found that fulvestrant can block this progestin-induced sphere assay. We also tried onapristone, mifepristone, and villaprisan, which are the sort of the anti-PRs that you can acquire. And fulvestrant does block this pretty well in green, suggesting that we do have this ER-PR dependency. And interestingly, and I think this is really fascinating too, even though fulvestrant blocks, tamoxifen does not. So fulvestrant is a degrader. It's going to degrade the ER and make it a lot less abundant, whereas tamoxifen is a SERM. It's going to bind to the ER and block its activity. So we don't need the activity. We need the scaffolding action. So that's why tamoxifen doesn't seem to work, whereas fulvestrant does work. And she also found that this kinase inhibitor, palbociclib, works really well. And it turns out that CDK sites are very abundant on the PR. There are 8 sites, and we'd like to look more at that in the future. And then finally, this really cool story that's emerging. We think that PRA is bad, and tumors that have highly abundant PRA should probably be identified as high risk. And so Noelle has started looking into this long read sequencing idea, where you can actually measure the transcripts for the PR isoforms. And so this shows you the PR gene and its exons that give rise to PRB and PRA. And then there's this PRC, which is actually expressed in the uterus, and we don't really see it in our models. And so Noelle performed the long read sequencing, where she validated the expression of PRA and PRB by Westerns, along with Carol Sartorius's lab and Jess Finlay Schultz, who did these Westerns in these PDX models. And then she went ahead and showed that the A-rich cells have A, the B-rich cells have B. These PDX were A-rich, and the tamoxifen resistance ones were A-rich. And so with that validation, she now has been collecting tumors, patient tumors, and she has this set of 5 where she has paired normal and tumor. And in every case, they are A-rich, almost every case. Some of them are way more A-rich than others. And we don't always have a paired example. So here we have normal, which is kind of a 1-to-1 ratio, and the tumor, which can have an A-rich phenotype. And so it's possible that you could use this as an easy way. You don't need much RNA, an easy way to look at isoforms clinically, and nobody does that. They only look at total PR. And there's no really easy way to do that with antibodies, because of course, all of the PRA is identical to the PRB. And so that the antibodies in the tumors have been really controversial. So this could be a really nice clinical tool someday to say, if you have a super A-rich tumor at diagnosis, you may need a little more tough treatment to block those stem cells and maybe palbociclib, right? And so it gets me back to this dream complex that we think that PRs can mediate this durable quiescence and dormancy and maybe late recurrence, and that's mediated by the phosphorylated PRA. And we think maybe palbo could work because we know CDKs feed into this path, into these the regulation of the proteins in dream. We can use fulvestrant to eliminate ER, and maybe some things like PR protax in the future, or something like that, to get rid of PR degraders. And there is a company which has PR protax. They work incredibly well in mice, and they are in Switzerland, and we'd love to see clinical trials with those protax. But we think that PR can drive, when you inhibit breast cancer growth with endocrine treatment, but you don't kill, you're driving cells into G0, and then that's allowing progesterone receptor action to manifest as supporting those G0 cells. Okay, and so this is just proof that PR is in that complex from Laura, where she found that PRA, these progestins could increase stream here and decrease this complex. So therefore, supporting cells in G0 and blocking cells from entering the cell cycle. And you can see really nice IPs with PR in the dream complex proteins. So just really beautiful work that was published in ovarian cancer models, but we'd like to move this into breast to really prove that PRs can be mediators of the change in cell fate that can give you this dormant phenotype, and that we need to target that. And so in summary, here's back to our big ugly tumor, and we target the estrogen receptor very effectively at first, but then if we don't kill those cells, they can come out and still mediate the expression of PR. And with all of those mutations that activate kinases, the PR is phosphorylated and can really potently support the stem cell compartment. PRs are fully integrated with the estrogen receptor and also the insulin receptor, which I didn't spend too much time on. Phospho-PRB drives the endocrine resistant states, we think, in collaboration with ER. PRs can drive numerous non-proliferative cancer hallmarks, namely PRA supports stemness, and these are targetable biomarkers in ER positive cancer that can predict metastatic potential, we think, and that clinical measurement of these isoforms is on the horizon and may identify high-risk patients with A-rich tumors who might benefit from things like palbociclib or other targeted therapies that could be blocking the stem cell compartment. And then these are all the players, lots and lots of people: Julie Ostrander, Laura Morrow, Kaylee, I mean, just so many people. Alpay has been invaluable. My lab is listed here, and then the cores. And these are my sort of early on PR dream teamers. And, you know, of course, too, has had her lab a couple of years and is still a valued collaborator in a lot of the stem cell studies. Amy is faculty at Adelaide now and still working on steroid receptors. Caroline is sort of the behind the scenes energizer bunny of the lab and has done a lot of these experiments so beautifully. And I apologize if your name wasn't on these. And then, of course, Noelle has just been a force in the lab for the last 2 years and has really collaborated with everybody on all the omics and analysis of gene sets along with Alpay. And so, and also has really creative ways to look at the PR isoforms, which is sort of, I think, revitalizing the field, actually. When we think we know everything about these receptors and then we see these big things. And I really think this PRA signature, it just blew me away. I was really surprised by it. Okay, and then I think that is it. This is, oh, my little shithead Momo. And he did meow every half an hour all night long last night for the first time in his life. I don't know why. I think he wanted to go outside, but every half hour it was meow, meow. I was just like, oh my God. But I just wanted to thank all of you for what you do every day. I know in this environment, we sometimes don't feel valued. If you are in the trenches in the lab, I highly value you. And if you're seeing patients also, thank you so much for that. I have found over the last year that everything we do is so important. And I don't want to cry, but thank you so much for that. And keep going in this weird environment. All we can do is focus on what we love to do. And you're not alone, and we can get through it. We're going to get through it. It's going to be painful, but hopefully we can look back at this someday and say, I lived through the craziness, right? And we will keep going. So thank you very much. You can see the title of this paper is the early seeds of metastasis that showed that the cells that escape the tumor early have to have PR and Wnt and rank ligand. And it wasn't my lab. But Momo ate it when I was writing my grant.
Carol, that was fantastic. It's so cool to see the history that you've carried over the last. I'm just gonna not say that number because it's embarrassing.
Yeah,
But,
I'm old.
you know, they say that every good answer generates 100 questions, but I'm gonna keep it to 3, maybe simple ones. Can you do differential staining with an antibody that recognizes the N-terminus of PRB, and then an antibody that recognizes total to infer? Is that possible?
Yeah, people have done that in the literature, named Sandy Haslam, who has retired, but they would do a total and then subtract the PRB. But it was pretty cumbersome, and there was a lot of controversy in the field whether those antibodies were working well or not. Some labs get it to work for PRA and PRB. Namely the group in Sydney, that's excellent, Denny Graham and Christine Clark. But when we have tried to repeat that, Carol Sartorius, who's also fabulous, tried to repeat that in her lab, she couldn't get that to work. So they don't, it's kind of slide of the wrist.
It's a reagent issue though.
It's been really difficult to do.
Yeah. So have you thought about the fact that when you're looking at these cell states that you're modeling, that in the 3D, you know, you showed us that picture of the 3D tumor and all the stuff that's happening, right? Do you think that all of these states are coexisting?
Probably, yeah.
Yeah.
Yeah. Heterogeneous.
Yeah. So is there any way to look at this on a spatial or single cell?
Oh, sure.
Yeah.
If I had $1 million,
I'll
I'd buy you a green dress.
You take checks. Yeah. What do you think these pathways for ER/PR are doing in the immune system?
Oh yeah, so Christy Hagan, who was my postdoc, works on that. They do a lot to recruit cells to the tumor. We showed, and it didn't repeat for weird reasons because the mice. So we got these AIB1 mice from Miles Brown, which is SRC3. So that's a coactivator that's highly overexpressed in breast cancer. And they get ER positive tumors that can be tamoxifen resistant. So we showed in that model just a 10-day gavage of antiprogestin, blocked macrophage infiltration very well, blocked Ki-67 very well. And the mice take a long time to get tumors, so we didn't carry that out. And then what happened to those mice, and Miles never told me this, is what happens over the breeding process is even though they genotype fine, they don't produce litters. And then what happens is you don't get expression of AIB1, and you don't get that high expression anymore. And so they crap out, and so you get very variable results that are very expensive because you have all these cohorts. So we just did the short-term experiment with gavage, and we showed tamoxifen and onapristone could block the markers for the macrophage infiltration, and the Ki-67 was much less, and they worked similarly, tamoxifen versus onapristone. But we just never published that because we couldn't reproduce it. And then when I talked to Miles, it's like, oh yeah, those mice crap out. And they got 2 cancer research papers. They had to rederive the mice several times to get those papers.
Hey, Carol, do you think that like healthy breast tissue has both isoforms of PR, or is this like a cancer specific event?
Yeah, okay. So normal tissue in the uterus or breast or brain or wherever has 1-to-1 ratio. And a very early event is the lopsided expression one way or another.
And I guess I was wondering, is there any knowledge, like idea, like what drives expression of these isoforms? Like anyone know what factor could be regulating?
I would love
this?
to know that.
Yeah.
So there are 2 promoters that we call a distal and a proximal promoter. It's not really clear if those promoters regulate A versus B. Very few people work on this, but there's a lot of stuff in those promoters. So not only EREs, it turns out that PRA expression in animal models at least is highly dependent on estrogen. But PRB expression is much more promiscuous. And the only one I know is Claudia Linari has shown that FGF2 can increase the expression of PR. And Caroline Deep in my lab showed that progesterone increases the expression of PR. So just like with glucocorticoids, which kind of auto-regulate their expression, progesterone receptors, which are highly functionally redundant to GR, they can do that too. And they do that with SP1, AP1 sites. So there's a lot of sites that could be driving this. I think what happens is you probably have this tumor microenvironment that gets inflamed, and then you have all these cytokines, and then you start regulating those promoters a little differently with other things besides estrogen. And then things get all hairy, right? And if that's regulating the cell cycle, then you can see the cell cycle starts to get wonky over time. And I think it plays a role in development of the tumors at that point in time. I never thought it would play a role in more advanced phenotypes like the stemness phenotype. I never thought that would happen. I mean, my lab used to be all focused on early events early in my career, and now we're all focused on advanced. Doug.
Carol, have you ever had a chance to look at the PRA and B ratios against the established, like Oncotype or Mammoprint? Because at least in Oncotype, PR levels are on the good side. A high PR level will give you a favorable Oncotype score. Now, everybody also knows the Oncotype score doesn't tell you the whole thing, and they never deconvolute the score. And I don't even think PR is in the 70 gene assay. So anyway, have you had a chance?
So we're just starting to get there, and Susan Schmidt in my lab, she treated cells with estrogen and just looked at PRA versus B, and the PRB signature in 2D is protective, very protective. And if you have the estrogen driven growth with PRB, it'll be less than PRA. And so I think the PRB rich tumors are going to be inhibited. They're going to be the dog that's pulling back on the ER. And then as we get to cells that just have PRA or a lot of PRA, that is supporting an exit to the cell cycle. So it should be less growth. It should be reduced Ki67. It's supporting a change in cell fate to the G0. And that's what I think people can't wrap their head around because we're so stuck on proliferative markers. But so Noelle probably needs to do that now that she has the 3D data, is now go back and look at these gene sets, the Oncotype DX and the Mammoprint and all of that.
There are a variety of ways to extract those assays out of RNA-seq. Just let me know. Laura Van Veer, who does this, is doing it on the iSpy data set. So you can get, even though you don't have to use the same platform. They figured out how to do it, what the model is for RNA-seq.
Okay, I think we're the only people that have the 3D gene signature at this time. It's never been published, and it's very different from the 2D.
I just want to reconcile two parts of your talk. So at the beginning, you had mentioned that luminal A tumors are enriched for PRA, whereas the luminal B tumors are enriched for PRB. And then at the end of your talk, you established a case that PRA is the bad actor. So how do those go together?
Yeah, so Claudia Linari published that finding using antibodies and a very limited tumor set. And so I had that in there because of that paper. But it's not been validated outside of that. And, like I said, I was really surprised to see that PRA is nasty in the 3D gene set and a driver of the stem cell compartment, a potent driver of the stem cell compartment. So I think that, as these tumors go to luminal B, they have a lot less ER and PR and a lot less PR and often undetectable PR. But then I showed you in all those models, it's still able to mediate major tumor biology, even though there's very little of it. So that's something we're still, a lot of the genes are secreted factors, so you could kind of see how that would work. And if the stem cell compartment is also quite small, maybe those are the cells that are supporting the stem cell compartment, not really driving proliferation at all. And so everybody says they're tumor suppressors, which they are because they're inhibiting cell growth. And what I'm trying to say is yes, but it's a teeter-totter, and now you're supporting dormancy. You got to kill them. So how are you going to kill them, right?
Carol, quick question. When you were having that CTCs kind of reduced because of PGRA, right?
Mm-hmm.
I was wondering, have you looked in, is it important to play a role in initiating EMT?
We're looking now, and we're doing that in collaboration with an EMT expert. So we're just now starting to look at that.
Because I also know that there are some data sets of CTC cells from breast cancer patients or like EMTs transitional signature.
Yep.
And I wonder whether you actually can correlate the racial changes.
Yeah,
between
that's where
PGA and
we're
PGB.
heading, where we're heading with things like TGF beta. That's a really good mediator of EMT. And GR, I suspect it's a triple negative. It's going to be GR. And it's a very similar phosphorylation GR story. And then possibly in luminal, it would be PR. So that's really exciting. So that's kind of a direction we'd like to go. And Claire Keaty, who's a new student in my lab, is going to work on that.
All right, let's give one more round of applause for a great presentation.